Greg Matlashewski, Ph.D. professor, McGill University, and former program leader, World Health Organization. His research has been influential in the field of neglected tropical diseases.
Hello, my name is Greg Matlashewski. I'd like to share with you some of the research we've been doing on a neglected tropical disease known as visceral leishmaniasis. Now, this is a picture of a village in the northern part of India in the state of Bihar. The state of Bihar is actually the poorest state within India, and there's a population of 90 million people. This state has a number of infectious diseases, but among the most important neglected tropical diseases is visceral leishmaniasis that kills thousands of people every year in the state of Bihar. Now, why are we so interested in the state of Bihar? Well, if you look at this next slide, you can see that this is the only state, in fact, this area here, where you have a lot of cases of visceral leishmaniasis. This is where the majority of cases are occurring, is just in this northern state, which borders Nepal here. In fact, most of the cases are just north of the river Ganges. Now, in fact, this area has 50% of the cases on a worldwide basis. The other 50% of the cases occur in sub-Saharan Africa and some parts of South America. So this is a real hotspot for the disease. So if you're going to start to try and control a disease, this is the best place to start. So this is where we've been working on visceral leishmaniasis. Now, there are good treatments for this disease and there are good diagnostics for this disease. The problem is that with 90 million people, it's just so difficult to have good surveillance to make sure the people who do get the infection actually get treated. So working when you have thousands and thousands of villages like this and people get infected, the vast majority of them don't know what to do when they have the disease. In fact, they don't even know if they have the disease. And many times they'll go to a village quack where they don't receive the proper treatment, and then it's too late, and they continue to transmit the disease to others. So how do you tackle an issue of surveillance when you have limited resources with such a vast number of population? Well, the way we've approached this, and this is work we've been doing in collaboration with the World Health Organization, has been to work with village ashes. Village ashes are women that live in the village and they look after the maternal health. Their major goal is to just look after the maternal health within the villages. But one woman has that responsibility within the village. And she knows what's going on in these villages. So if you work with these ashes, if you explain to them and train them that there is visceral leishmaniasis in this area, and if you identify a case, send them to the primary healthcare center where there are effective treatments and they can be treated for this disease. If they don't go, as I say, they will end up not being treated in time. So this has been a very effective program, and village ashes have been very, very receptive to this. And this has been now rolled out to large parts of Bihar to improve surveillance of visceral leishmaniasis and improve treatment of visceral leishmaniasis, improve the number of people who get treated for visceral leishmaniasis. Now, in addition to that, we've also been interested to understand how the disease gets transmitted within these villages. This is a village known as Tarawa. We worked here several years ago. In this village, there were 40 cases. The population of the village is about 1,000 people. There were 40 cases, and there were actually 6 cases just within this dwelling here. So what we've done is we've gone into these villages and we've done a serological survey. And the way we do that is you take a drop of blood, just a finger stick of blood from an individual. It's not painful, and the drop of blood is put onto a rapid diagnostic test, and the rapid diagnostic test actually is shown here. Here's a strip, and it will show you whether it's positive or negative, and we can determine how many people have antibodies. If they have antibodies, they've been infected, but the vast majority of people who get infected don't get the disease. This is common for many infectious diseases. Only about 10% of them actually end up getting the disease. And so we've gone into to determine how it's being spread, and particularly in households, to understand how it's being transmitted in households. This is a picture of myself. I've been involved with this study for several years, and I've been very fortunate to collaborate with wonderful people. This is Dr. Das, who was one of the collaborators from the Indian Council of Medical Research. These are other researchers as well who have been involved with the study. And what we found is that, as I was saying, the vast majority of the transmission is actually occurring in cases. Asymptomatic people don't appear to be transmitting the disease, but it's mostly the symptomatic people who transmit the disease. And this is good because now you know where to focus your efforts. You need to focus the efforts on identifying those cases. And this is what we're doing to, again, to improve surveillance. The other aspect that we're working on is the development of a vaccine. And this is work that's going on in my laboratory at McGill University, where we have a very strong molecular biology program where we can actually go in and genetically modify the genome of Leishmania. And so we, in my lab, we do the genetic aspect of this, of these development of vaccine, and we use Crispr gene editing. This is just a depiction here where you can use Crispr to go in and specifically alter chromosomes. And what we've been doing is we've been doing it in the Leishmania organism, where we go into the nucleus and we alter the chromosome. More specifically, what we've done is we used CRISPR gene editing. So this is a editing complex here. This is another editing complex where we go in and we cut out a gene, and we know which gene to cut out. We cut out only a single gene. There's 8,000 genes within the genome. We cut out one, and then we stitch a chromosome back together again. If we don't put it back together again, it won't survive. So we have to repair the chromosome, but the organism is then alive, and it looks exactly like the normal unedited organism, but this has now had one gene removed, and it grows very well in culture. You can see it. All of these are Leishmania grown in culture. They grow very well. And this culture now will no longer cause disease, but it will stimulate a protective immune response. And we know that because we've done experiments in rodents, where when we vaccinate rodents, we see that all of the rodents survive the infection, and the unvaccinated rodents, all of them die from the infection. So this approach of developing an attenuated strain has been effective in developing a vaccine. And the vaccine is now being produced by a company in India known as Geneva. Geneva is a major generic pharmaceutical company. They're very capable of producing this vaccine. India, in fact, is the largest vaccine producer in the world. And this is important because the vaccine is produced in India and it's going to be used in India for vaccinating these areas within Bihar where you have high levels of infection. And if it's shown to be effective in India, then the vaccine can be used also in other countries as well for vaccinating in sub-Saharan Africa and South America. So I'd like to end with this picture. This is a young boy who had visceral leishmaniasis, and he was brought to the primary healthcare center. He was successfully treated and cured. This is the mother who brought him in. As I say, there are treatments. The problem is to make sure that this disease stops being transmitted within these villages, so that there are much fewer cases. The majority of cases are children, in fact, so we want to make sure that this disease gets eliminated so that it stops, we can eliminate it from this part of the world and other parts of the world as well. And finally, there's a picture of the collaborators. Much of this collaboration has been done with the World Health Organization, with the Indian Council of Medical Research, and then I have a number of wonderful collaborators, particularly who do the immunology of the types of research that we do when we develop the vaccine, which I wasn't able to talk about. But with that, thank you for listening.